This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 241, recorded on April 29th, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, another TWiM.
How are you, Michele? Everything good?
Yeah, getting ready for medical school teaching, which unfortunately we have to do remotely. So recording all my lectures, which is sad.
Well, next semester, maybe you'll be in person, right?
Well, our course is in June. So
maybe in September you'll be back, right?
Yeah, I don't teach in September, but yeah, hopefully.
Yeah. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How are you doing, Michael?
I'm doing well. I'm holding out, turning on the air conditioning. It's about 81 degrees outside, and the house is getting a little toasty. And I said I want to not turn on the air conditioning until May this year, and hopefully I'll be able to withstand the next 2 days.
I see you have your window open back there. They could see
I do. We have a nice breeze coming in, and
right.
I'm working from home today because our institution is embarked on a major construction initiative. So outside my office, I have the beeping tractors, and I didn't think people would appreciate the front end loader going beep, beep, beep back and forth as it moves dirt. So they're building a new pharmacy school.
That's why Elio's not here, because he has some construction in his apartment. So he gets the week off.
Yeah, we have to quiz him as to what color he's going to paint his walls and what color carpets he got.
Right. All right. So we will start off with a snippet from Michael.
And this actually was a snippet that was selected by Elio. So I'm going to try to channel Elio as I do this snippet. So the title of the paper is Inverse Regulation of Vibrio Cholera Biofilm Dispersal by Polyamine Signals by Drew Bridges and Bonnie Basler. They're both from the Department of Molecular Biology at Princeton and the Howard Hughes Institute. Drew is a postdoc in Dr. Basler's laboratory, and this paper was published in eLife on the 15th of April. And it's an open access manuscript, so you'll be able to follow along with us. Well, the title is the story, and the authors go through a tremendous set of what I will term bread and butter type experiments in demonstrating the veracity of the claims of their title. First, as background, they offer that Vibrio cholera undergoes repeated rounds of clonal biofilm formation and disassembly. This occurs on a routine, if you will, cycle of approximately 12 to 15 hours, where both the biofilm forms and then the biofilm deforms or decompresses itself, and the cells become motile again and move. Now, this concept is central to disease transmission, as Vibrio cholera alternates between its marine niche in these estuarine environments and in the human host. And the Vibrio cholera biofilm life cycle occurs in three distinct stages. First, a founder cell attaches to a substrate. Second, through cycles of growth, division, it begins to lay down that extracellular matrix secretion, the EPS, or as the orthopedic surgeons refer to it, the concrete. These biofilms actually make this extracellular matrix material that is really challenging to debride, and it's really the scourge of all biofilm infections. And then The third stage, as the biofilm matures under the appropriate environmental conditions, when they are detected, the cells leave. The biofilm actually disassembles itself through a process termed terminal dispersal. And first, I think it's only fitting that we discuss a pathogen in this year of global pandemics that's actually a bacterium responsible for a pandemic. And this microbe or bacterium has been responsible for 7 pandemics that we know of, commencing in the 19th century, where cholera spread across the world from its original reservoir in the Ganges River Delta in India to the subsequent 6 pandemics that have killed millions of people across all continents. The current or 7th pandemic started in Southeast Asia in 1961, reached Africa in 71, and the Americas in 1991. Cholera, unfortunately, is now endemic in many countries, including the most recent occurrence where its endemicity has arrived is, of course, in the island of Haiti. Now, cholera, as background for our lay audience, is an acute diarrheal disease that can kill within hours if left untreated. And when I say diarrhea, it not only fills the cup, but bring a bucket as well. In extreme cases, it can result in the loss of over 1 liter of fluid per hour, with extreme cases resulting in up to 20 to 30 liters of fluid loss per day in the affected individual. So you can well imagine that you just simply go into electrolytic shock because you lose your electrolytes from losing that much fluid.
Dehydration.
Oh, it's just horrific. But we have a simple solution to combat cholera today. It's called oral rehydration solution, which is a little bit of glucose, a little bit of salts, and it's really inexpensive. And the WHO has packets of these stockpiled, so if there's ever an epidemic in your local area, we can quickly deal with it. You also need
and also providing safe water.
Oh, absolutely.
Treated water to drink.
And you need to ingest about 100 million of the cholera microbe in order to really develop these horrific symptoms. And in fact, most cases are asymptomatic. So that's enough on the background of the beast. What we'll witness in this manuscript are the following type of experiments. And I thought I'd lay out the experiments that we are going to see. Since this is open access, you can have the opportunity to dive deep into each of these really elegant experiments that Bridges and Basler offer for us. So the first one is real-time biofilm life cycle measurements. Here we can literally watch the fruits of their bright field microscopy assay. And I'll put a video link in the show notes to one of their figures so you don't have to hunt, that enables them to witness a biofilm life cycle of Vibrio cholera. Now, you're not going to have to watch 15 hours of video. They compress the 15 hours into 3 seconds. And it's really elegant. 15 hours down to 3 seconds. So you can tell this was designed for the YouTube generation. The second batch of experiments are classic mutagenesis, the type of experiments I grew up on when I was a postdoc, making mutants, deletions, complementing those mutants, and a high contact imaging approach to identify mutants that fail to properly disperse, because their signal that they're looking for is biofilm decolonization or biofilm formation, which then allowed them to characterize the functional groups, the pieces parts that were interpreting whether it was time to make biofilm or whether it was time to go. And the way they did this is they asked the question, what were the pieces that were involved in signal transduction? Secondly, what was the signals or the genes that were associated with this matrix degradation, or getting rid of the EPS or slime that we associate with biofilms. And finally, cell motility. Because remember, Vibrio is a very motile microbe, has a polar flagellum, and where disruption of the production of gene products enables the authors to test their hypotheses that these classes of components act in sequence to drive biofilm dispersal. It's a familiar story to folks who think about complex microbial behaviors and gene regulations. So first up, there needs to be a cue or a signal that it's time to go. And here they ask what cellular components detect the cue. Now, if you know anything about biofilms, it's all about matrix or EPS. So the next event is the activation of gene products for this matrix digestion. So thinking about the cue, we know here that cyclic di-GMP regulates biofilm formation in many bacteria, including Vibrio cholera, in which low levels of the cyclic di-GMP levels correlate with motility, so no biofilm, and high levels of cyclic di-GMP promote the surface attachment and matrix production, and thus biofilm formation. That's an old observation that they offer to us in the introduction. This is where they bring their third type of technique or experiment to bear, and that's the use of the reporter cassette concept to measure cytoplasmic concentrations of the cyclic dinucleotide GMP, which, unlike the prototypical second messengers of the single cyclic nucleotide cousins, those that you likely learned about when you first mastered the lac operon, cyclic AMP and how it would interact with catabolite activator protein to effectively become a transcriptional activator. That's the sort of thing that is likely happening here is that this cyclic nucleotide, albeit a dinucleotide, correlate then with this behavior that they're using as their readout. So again, low levels of cyclic dinucleotide GMP correlate with motility, and high cyclic di-GMP correlate with surface attachment and matrix production. But that's the second messenger. So what's the first signal? And how does the first signal generate the second signal? We know in the lac operon, lactose is that first signal. It interacts with the lac permease, creates allolactose, and then the allolactose de-represses the repressor protein, etc. So what's actually going on here? Well, they describe polyamines. These are essential small molecules derived from amino acids, and they offer that they were likely produced by the last universal common ancestor. Different organisms make different polyamines, and they are involved in these biological processes ranging from stress responses to signal transduction to protein synthesis to the public good factor siderophore, which brings in the iron. So intriguingly, biofilm alterations that occur in response to a given polyamines vary amongst species. So in the case of Vibrio cholera, norspermidine, a rare polyamine produced by the Vibrionaceae family of Proteobacter in the order Vibrinales, and other select organisms promote biofilm formation. So it's known that, and this has been known since about 2005, that norspermidine promotes Vibrio cholera biofilm formation. In contrast, the nearly ubiquitous polyamine spermidine, which differs only from norspermidine by a single methylene group, is not produced at substantial levels by Vibrio cholera, and it represses Vibrio cholera biofilm formation.
That was amazing to me that such a modest change in the structure of the molecule could completely change the lifestyle of the population.
Well, we're witnessing that sort of today, Michele, in our education on the fly with spike protein and coronavirus. I mean, we're seeing how single amino acid change can do things. And that's the fourth type of experiment that they present data from, where they measure the intra and extracellular polyamine concentrations to define the roles that norspermidine, which encourages film production, and spermidine, which instructs the biofilm to stop things and to get ready to move on, play in the controlling of this biofilm life cycle. All of this leads to their overarching hypothesis that Vibrio cholera detects norspermidine as a measure of self, and spermidine and spermine as measures of others to assess species composition of the surrounding microbes in their local niche. They use the term vicinal community. And again, this is a paper from Princeton, so they can use words like vicinal because it's a good SAT word. So I'm going to get letters from the Princeton folks, I know. Vibrio cholera funnels that information internally to determine whether or not to make biofilm. And Vibrio cholera produces intracellular norspermidine. However, and this was what I thought this was going to be a classic quorum sensing paper where, you take a low molecular weight product, you throw it out into the environment, it comes back, it turns on a bunch of genes. I thought it was going to be unremarkable. But this norspermidine has laser-like focus to control gene expression and coordinating this complex opera of biofilm formation. So norspermidine has not been detected in cell-free culture fluids from laboratory-grown strains. Thus, if non-spermidine does indeed enable cholera to take a census of self, it is not via this apparent canonical quorum sensing type mechanism where one cell makes a quorum molecule, it goes out, and it waits for it to come back in before it turns on its regulation. Now, all of this is summed up beautifully in their first figure. And they use this first figure in many of their figures. So if you grasp this figure, digesting the data in the subsequent figures becomes really easy. So here's where they offer you all the pieces. And they offer that when you have high norspermidine concentrations, you stick to something called the norspermidine sensor, NSPS. And like all genetics papers, it has lots of abbreviations. So I know this will drive the people driving their cars crazy, so I'll just try to call it sensors so you can sort of follow along with us. So the norspermidine sensor binds the norspermidine and it interacts then at the inner membrane. So norspermidine sensor is in the periplasmic space and it interacts with a membrane protein that they term MBA-A, that I'm just going to call MBA. And when it interacts with MBA, this is how the MBA then communicates to the polyamine sensing dinucleotide GMPs. And they go through great lengths to illustrate this, but you'll get the idea of the model and the player simply by watching their first video of their bright field microscopy. And I'm just going to walk us through that. So what the video shows is a representative time-lapse image of biofilm life cycles. And they're organized pretty rationally. We have wild type. We have a knockout of the inner membrane component, MBA. Then we have a knockout of an inner membrane receptor called delta pot. And then we have the double mutant. And what they do for us is they have one column that they treat with water. They have one column they treat with the biofilm forming signal norspermidine at 100 micromolar. And then the last column is 100 micromolar spermidine. So recall that the process of wild type biofilm life cycle, just focus on the upper leftmost image. In their 15-hour compressed to 3-second time lapse, you literally will witness formation and destruction of the biofilm. What you see from this and then the subsequent deletion mutants is effectively illustrating their title. Norspermidine actually coordinates this biofilm dispersal, or inversely controls this biofilm dispersal process. And so it's really pretty cool about how they do this. And when they get to the mutants, you then begin to really appreciate this transduction of what's going on. So what was most impressive to me is the bug out signal column, the spermidine. And what you see in that video is that the biofilm never really gets started. It tries, but fails to form a film. And as they walk us through the very detailed genetic analysis of this, looking at concentrations, this is where I think the first author, Drew, in one of his Twitter responses says, these signals are laser-like in focus. And it really highlights what is going on in this process. So bottom lining this, norspermidine and spermidine inversely regulate the Vibrio cholera biofilm dispersal. The next bit of the story was by what mechanism was this norspermidine, spermidine information encoded being translated into alterations in matrix gene expression. Here their hypothesis was that the intermembrane receptor, MBA, and or POT mediate changes to cytoplasmic cyclic dinucleotide, GMP. And it was through this modulation of this classic second messenger that changes to matrix degradation occur. If you're interested in learning more about cyclic dinucleotides and how they're involved in gene regulation, I put in open access Nature microbiology reviews on the subject into the show notes. So here's where the two primary cues come back in. To make a community is by norspermidine, and the time to deconstruct is signaled by spermidine. When provided alone and at a concentration above 10 micromolar, norspermidine and spermidine strongly increased and decreased, respectively, the di-GMP reporter activity. At the limit relative to wild type, norspermidine increased the reporter output by 60%, while spermidine only reduced reporter output by 25%. There's more to the story, and there's a second video where they begin to dissect which domains are important. But since this is a snippet, I'm not going to go into all that detail. And since it's open access, I'll let you take that on your own. And so what we know so far is that norspermidine interacts with the periplasmic sensor NSPS. And when bound, it communicates to the binding of MBA that results in it communicating with the cyclic dinucleotide synthesis machinery that in turn leads to matrix production. However, in MBA, there's a domain, and it's different from the canonical domain of GGDEF, and it's now SGDEF, which effectively renders MBA inert, but it's still able to transmit a signal to norspermidine telling it to upregulate the synthesis of the dicyclic nucleotide. They postulate that MBA is a bifunctional protein capable of both diGMP synthesis as well as degradation. So they ask, what's in it for the Vibrio? And here they speculate that because MBA can inversely alter this level of second messenger. And those of you who have read a lot about second messenger gene regulation knows that this is a common theme. The second messenger, in response to these two discrete cues, will effectively enable the Vibrio to more rapidly and with greater magnitude change the concentration of the cyclic dinucleotides than if two receptors were required and they were somehow separated. And again, they point out that this is not without precedent. And you can read more about this and the discussion. And finally, they offer some caveats. The observation that laboratory wild-type Vibrio releases little non-spermidine, which was really the puzzle at the beginning, is in part due to the fact that POT, that periplasmic factor, mediates reuptake. And this suggests that this system does not behave like the canonical quorum sensing pathway. However, they offer that this norspermidine is secreted by Vibrio under some environmental conditions or by other Vibrios, and that cholera that can then detect the norspermidine that has been released from biofilms that may have been destroyed either by predation, phage, or toxin-induced cell lysis. So specific.
Right. To me, that was like key to understand that the norspermidine, if the cell is healthy, they may release some, but they take it back up. But if the cell is sick, it's under some kind of pressure or danger from predators, it can't do that. And therefore, accumulation of that norspermidine is a stress signal to the rest of the population. I thought that was really lovely.
And the other thing that they don't offer is, remember, biofilms are, for the better part, viable but non-culturable cells. They're effectively not very active metabolically. It's a mixed population, right? There's a lot of division of labor within that biofilm. So Absolutely. You're right. A lot of them are, but others are doing the work of the community. So, and I would recommend that you read the entire paper to pick up the nuanced arguments, but we'll offer you what they leave us with in the last paragraph of the discussion. And I think it's only important to conclude with this, that this microbe that has been responsible for at least 7 known pandemics, and cholera has been around since recorded history, is it shows us the elegance that fitness strategies of pathogens have developed over time, and the elegance in the regulation of Vibrio cholera biofilm formation is through 4 principal means. The first is starvation. The second is the classic canonical gram-negative quorum sensing approach via the Chi-1 and AI-2 signaling pathways. A recently identified DBFS and DBFR system with an unknown ligand, in addition to this 4th system of polyamine control via the norspermidine sensor and MBR transmitter. What I take away from this, it's no wonder Vibrio has 2 chromosomes, and it's remarkable it's only caused 7 pandemics.
Well, it makes sense that it takes a lot of energy on the part of the cell and the population to make a biofilm, but there are going to be times where you need to get up and find richer territory elsewhere. So they've got many mechanisms in place to make that decision collectively.
And I think the take home is it knows where it is. It's continuously sensing its environment.
Constantly looking around.
Yeah.
Is it still okay? Is it still okay?
Do we understand the signaling that goes from the spermidine to dispersal? That's not
Yes.
the subject of this paper, but is that all worked out?
To some extent, and that's the stuff I glossed over that you can go into greater detail. That's
right.
the environmental decamping signals. I thought I'd get into trouble if I go into too much detail.
Yeah.
And again, I
think
was trying to channel elio, but wasn't too successful.
So you either have a dispersal of a biofilm or you have aggregation. There are two different things going on, right?
Right. Opposites.
Yeah.
Yep.
From one methylene substitution.
That's amazing. Yeah. It's interesting to me also that these two compounds are used because they have many other functions in biology, right? And here they are floating around in the water and being sensed to determine if stay or go. It's very interesting.
Yeah. It sounds like the norspermidine is only out in the water if there's been some cell lysis going on.
That's true.
Something's attacked.
Cool. Very cool. All right. And it's interesting. There are 4 different systems that are integrated in some way to make this decision, right? Like, as you just mentioned,
it's
pretty complicated.
Big decision.
Cool. All right. I like this little tweet here. Contrary to popular belief, polyamines are not boring. It's from Drew Bridges.
He posted that tweet the day after his paper went live, and he of course gives the URL to the paper in his tweet. So I thought it was really, and he's really right. He's spot on. It's laser-like focus.
I don't know. I would argue that no science is boring. You just need to look at it in the context, and see what it's doing, and then it's not boring.
Right. It becomes more and more interesting the more you know.
Yeah, precisely. Yeah, if you just look at a molecule, you may, what's so interesting about this molecule? But what does it do? Then it gets interesting.
Yeah.
All right.
You're gonna tell us about an age-old phenomenon. When you got a virus, you don't feel like eating.
I want to understand this more.
Well, we're going to find a little bit more about it. This is a paper in MBio. Respiratory viral infection alters the gut microbiota by inducing inappetence. So we have another Princeton word, Michael.
That's
there
true.
You go.
We have 2 Princeton words today. Maybe that's the title of the show.
Well, I beg your pardon. This is coming out of Imperial College of London.
Oh, it's even worse.
Yeah, Imperial College of London and the Royal Brompton and Harefield NHS Trust. The authors are Helen Groves, Sophie Higham, Miriam Moffat, Michael Cox, and John Tregoning. And, we did actually years ago, Michael, back in the day when Joe Handelsman was with us, we talked about a paper where the gut microbiota influenced the outcome of influenza virus infection in the lungs of mice.
Yes.
So there's talking going on, and this is going to explore it even further. And of course, we have a virus in it, so I feel a little more comfortable here. We have 2 viruses actually. And as everyone knows, we talk often about the microbiota that we have and the contributions that it makes to our health, and which we learn more about every day, of course. Many of these contributions have long reaches. So what goes on in the gut can affect other organs because things like short chain fatty acids can be made and dispersed among the organism. And one of the interesting aspects is how the gut microbiota influences the outcome of respiratory disease. Changes in the microbiome can influence host immune function, and that can determine the outcome of a respiratory virus infection. For example, if you feed mice a high fiber diet, they make more short chain fatty acids, and they have less lung damage and better survival when you infect them with influenza virus. Another reason to eat more fiber.
Yeah.
Oh, yes.
Gosh.
And it's not so bad. You just get some good cereal with fiber, and that's what I do every morning. Forget the bagel. There's no fiber in bagel, folks.
No fiber.
And anyway, if you're not in New York, the bagel's probably not very good. Now I'm being a little snobby, right?
You are indeed. But you are accurate. You are accurate and snobby at the same time.
So have that fiber cereal. And so that's influenza. There's evidence in other systems that, your microbiome can influence the outcome of respiratory infection. Now, this paper actually looks at the opposite. what a respiratory infection can do to the gut microbiome. There's also a lot of evidence about what's going on here, and this group has done some of it, and others. Lung infections can influence the abundance of various microbes in the gut, but they don't actually know why, although different viruses have similar influences on gut microbiota. So the idea is maybe there's some common mechanisms involved there. And one thing that they got interested in is weight loss after infection with either influenza virus or respiratory syncytial virus, a pathogen, particularly of young children. And one which we've talked about a lot on TWiM recently. We're actually going to do another paper tomorrow about respiratory syncytial virus.
So let me ask you this question. In RSV, do the children who get RSV lose weight?
I don't know. It's a good question. I should know, right? But,
I don't know. You can ask the TWiM team tomorrow. Maybe they know.
Or I'm not sure. What we're going to learn today is they probably do.
Yeah.
Certainly mice are going to lose weight, as you will see. Whether it happens in people, and the thing is, it's quite a lethal infection in young kids. So maybe weight loss is associated with it. The kids are at the age 6 months and so forth. They're breastfeeding, I suppose, or having formula. So I don't know if that's been studied. Good question. So that's what they're looking at here. Let's look at respiratory virus infection, the gut microbiome, and food intake.
Could I interject? According to cdc.gov, one of the symptoms of people with RSV is a decrease in appetite.
Yeah. So if you don't have appetite, you're going to lose some weight.
Yeah.
Yes.
Which can be a good thing, right? Depending on how far it goes. Okay. So in their first set of experiments, they infect mice with respiratory syncytial virus. And I have to say, they put a lot of virus into the mice in order to get effects, because this is a human virus into mice, and that's not unexpected. And so, as they say at the end, all of this, we don't know what it means for people, but it may provide some clues. If you infect mice intranasally with the virus, they lose weight after the first day, and then between days 1 and 4, their weight stabilizes, and then they start to lose more weight after day 5. Eventually, 15 to 20% of their weight. It's a lot by day 7. So, the controlled mice are PBS inoculated. They're fine, obviously. And I noticed that the weight loss it goes hand in hand with food consumption. You have to now measure what the mice are eating. Normally, we throw mice in a cage with a whole lot of food and we don't measure it, but you can measure it, and that's what they do. And this is very interesting. I didn't know this. The average amount of food consumed by one uninfected mouse is 3 grams a day. And the infected mice eat less. And at the lowest point, they're doing 0.5 gram a day.
Wow.
Of consumption. It depends how old they are. How old they are, right? Let's look it up and see. Let's look up the weight of the 3-week-old mice. That's about weaning time. Okay, so 10 grams at 3 weeks. 4 weeks, 16 grams. Not 200, Michael. That's like a rat, I think.
Okay. Mm-hmm. Well, I've been dealing with microbes my career.
They're eating a significant percent of their own weight each day.
They eat a lot. I mean, we have cages with the solid food on top, so they have to kind of reach up, and it goes down. You got to keep feeding them. We have big containers of food in there, and you have a scoop, and you put it in. Anyway, so these mice lose their appetite, and that's as they say, respiratory viral infection induces inappetence, and so they have less appetite and they eat less, and then they weigh less. So it's all connected, right? Infected, they lose their appetite, they eat less, and they weigh less. So now we're going to figure out maybe what's going on here, right? Why would a respiratory, and we've all felt this, right? You have the flu, you don't want to eat, right?
Nope.
Yeah, and is it helping the host or is it helping the pathogen? That's a
good question. I don't know that we will have an answer, but one can imagine that it's the host defense mechanism, right? It could be.
Starve out the pathogen.
Yeah. Okay. So then they say, what does this do to the microbiome, the gut microbiome of the mice? So they first do this outside of infection. So they restrict access to food so that the mice will lose weight at a similar rate to when they're infected. And this is very interesting. To do this, you have to put one mouse in a cage, right? Otherwise, you don't know who's eating what.
That's right.
So, and they say, but mice are social, and if you isolate them, it may upset their gut microbiome. So they have to do a control experiment where they
chicken and the egg. The chicken
they
and
just
the egg.
Put single mice alone and give them all the food they want, and they don't stop. Their microbiome ends up being the same. So that's a great control. And I didn't know that. I didn't know mice were social.
I think we found that during the pandemic with people too. They can be alone and they still eat just fine.
When I was in Sweden a couple of years ago, I learned that in Sweden, you're not allowed to have just one horse. You have to have two because one horse gets depressed by itself. Right? They need to have a friend to chill with.
Well, okay, I learned that fact watching Mr. Ed. He had Wilbur.
No, I didn't remember that, Michael.
You didn't remember Mr. Ed, the talking horse?
I do remember Mr. Ed. I don't remember him being with anyone, though.
He was with Wilbur.
Okay.
Wilbur.
So anyway, the result is that if you they do the experiment. They give mice less food to restrict their intake. The mice lose weight. They lost about 15% total weight by day 3. And then they collect their feces and look at the microbiome by sequencing. And they look at what they call beta diversity, which is the variation in the communities between the two samples that they have here, either fasting mice or not fasting mice. And they say at the phylum level, the fasted mice have a higher abundance of bacteroidetes and a lower abundance of firmicutes compared to the control mice. And at the family level, the major change was an increase of a family called Muribaculaceae. And they say this pattern is very similar to what happens after infecting mice with RSV. Because remember now, we have no virus in these experiments. We're just starving the mice. And you get a change in diversity, which mirrors, it makes sense, right? Because the virus is just simply making them lose their appetite. So it makes sense that you would have the same changes, right?
Right.
All right. So why do you lose your appetite when you have a viral infection? Well, a viral infection induces an immune response. You make a lot of soluble cytokines, right? So you can imagine them traveling to other parts outside of the respiratory system, parts of the nervous system that controls appetite and so forth. So they look at cytokines. And the first one they look at is tumor necrosis factor, which is a classic pro-inflammatory cytokine induced by many viral infections. And it can damage cells. It will damage a virus-infected cell, for example, as a way of restricting infection. So they actually give the mice recombinant TNF-alpha. They put it intraperitoneally. And by the way, this is a cytokine that goes up after viral infection. That's why they're looking at it. And previously, people have shown that if you block TNF-alpha during RSV infection, it reduces weight loss. So that's the connection here. So they look at it.
And it used to be called cachectin after it causes cachexia, loss of appetite.
That's right. So they inject, actually what they're doing is they're injecting a monoclonal antibody against TNF-alpha into the mice intraperitoneally to knock down the levels of TNF-alpha in the mice. And then they take fecal samples different times. So again, no virus here. We're just trying to figure out what's going on here. And the mice treated with the antibody. So remember, these are monoclonals. So they're not going to last forever, but they will reduce the level of TNF-alpha for a while. Those mice didn't lose weight through the first 5 days of infection compared to mice treated with an antibody, but not an anti-TNF-alpha antibody. And then after 5 days, that effect seemed to go away. They began to lose weight more rapidly. Now, these mice, as you might guess, have more virus in their lungs because one of the functions of TNF alpha is to reduce viral loads, and so that makes perfect sense. And again, they look at the microbiota in the gut, and they get these changes in diversity that they saw before, induced by virus infection. But it's not as altered as they see during infection. They get the similar changes, but they're of lower magnitude. And so it's not, there must be more than just TNF alpha involved in changing the gut microbiome here. Okay, so they then asked, if you give mice TNF alpha, what's going to happen? And this is in the absence of infection. So they give mice TNF alpha and they lose weight, and they stop eating. They lose their appetite and they lose weight. So maybe TNF alpha is part of what is doing this. So infection induces TNF alpha, makes you lose your appetite. And how that happens is a great question. We don't know. And then it changes your gut microbiome. All right. So that's the idea so far. Let's see here. Okay. So the conclusion is TNF may be involved, but because they see differences between TNF and infected mice, it's probably not the only driver of the loss of appetite and weight loss. They also asked, all right, could immune cells be involved? Because other people have shown that CD8 positive T cells, these are cytotoxic T cells that would kill a virus infected cell. Very important for limiting virus infections. Others have shown before that if you block them, you can reduce weight loss after virus infection. So they looked at CD8 cells. They can deplete CD8 cells in mice by injecting them with a monoclonal against the CD8 antigen, and they can then infect those mice. And what they find is that if you deplete CD8 cells, it prevents all of the subsequent weight loss. It doesn't completely prevent loss of appetite, though. It's very interesting. They still lose a little bit of their appetite, but the weight loss is prevented, actually. Now, the viral load goes up as well, just like when we depleted TNF alpha. If you deplete CD8 cells, the viral load increases because they're important for clearance of the virus from the lung. And nevertheless, when you deplete CD8 cells, even though it prevents weight loss, you still get a shift in the microbiome diversity in the time when you've removed these CD8 cells.
But they say that the mice, the anti-CD8 treated mice, only ate 1.8 to 2 grams of food per day over the last 2 days of the infection compared to the usual 3 grams. So could it just be substrate limitation that is accounting for this? Because
what do you mean?
That's a third of the substrate intake. The wild type is consuming a third more food, which would make more substrate, which would alter the microbiome.
But they're not losing weight, but it is.
But it would influence the microbiome. That's what you were saying is the microbiome, the changes to the microbiome. You didn't necessarily.
The CD8 depletion doesn't change their microbiome, interestingly. So, I guess that inappetence is not enough to do it, right? Maybe it's a magnitude effect. Is that what you're asking? Yeah. Yeah, it could be. It could very well be. They didn't really look into that, but that's possible. Sure. And in fact, if you infect mice with RSV and blockade the CD8 cells, it reverses the change in these bacterial families that we noted before as a cause of virus infection. So CD8 cells seem to be involved, and maybe they're making some soluble cytokine that is involved with that. Now, CD8s make a lot of interferon gamma. They actually look into that, but it turns out that is not playing a role. They do measure the effect of CD8 cell depletion on lung cytokines, and they identify a couple that decrease. And so those are topics for future experiments to see if those are, in fact, maybe the cytokines secreted by the CD8 cells are involved in inappetence and the fecal microbiome changes. The last set of experiments are to ask now, what is the change in the fecal microbiome doing to the mouse, right? And so, we know the fecal microbiome can make a lot of metabolites, right? So they look at that. They do fecal
and digest a lot of food that's eaten.
Yes. So all that fiber that you eat, the microbiome is digesting it because you can't. And so there are lots of metabolites. So they can take feces and do metabolomics, identify all the molecules that are there. They find, they do mass spectrometry, 803 detected biochemicals, and they could identify 705 of them. And they find that the profile shifts over time following infections. This is a lung infection with RSV. The metabolites produced in the guts are changing. And what they find is 23 of these pathways that are involved in making these metabolites are involved in lipid metabolism.
It's so interesting, isn't it?
Right? And they can sort all this out by, pathway analysis and so forth. So lipid metabolism, in particular, sphingolipid fatty acid metabolism pathways had some of the highest enrichment values following infection. So these products are going up. And among them are short chain fatty acids that go up after viral infection, which are known to be, they can be immunoprotective and reduce inflammation even. So maybe these are involved in ameliorating or changing disease in some way.
It could be part of our innate immune system.
It could be. That would be so cool, wouldn't it? Right? If your gut is part of your immune system that's responding to a distant viral infection.
And then if our change to a Western diet with high fat foods is inadvertently impacting this arm of the immune system.
That's right. You make you more susceptible.
Is that the reason we have so many autoimmune type disorders like Crohn's disease and irritable bowel syndrome and all of the others that people have been so desperately trying to decipher the initial trigger? It could be something as simple as diet.
Yeah, for sure. Because our diet is very different from when we first emerged as Homo sapiens, right?
Yeah.
We didn't have a lot of meat. It was hard to chase a
lot of fiber. A lot of fiber, though.
We ate a lot of fiber, and we exercised more because we had to catch our food.
So the last experiment they do is to ask whether this viral infection somehow makes the gut more susceptible to infection with bacteria. So they use the murine entropathogen Citrobacter rodentium, which is often used to infect the gut and look at pathogenesis. And they find no alteration in susceptibility or pathogenesis with or without viral infection. So the viral infection is not making the gut more permissive for good growth of this particular microbe itself. So that's good. But really the question is what so there are a couple of interesting things. I think what is making the mice eat less, right? We don't know the signal there. They think it might be something produced. Maybe TNF is one of them, but something else produced by CD8 cells. And what's the mechanism, right? Mm-hmm. Where are they going to reduce your appetite? We know parts of the nervous system are involved in regulating appetite, and maybe that's involved. So they say the anti-inflammatory drug indomethacin prevents interleukin-1 beta induced anorexia.
Oh.
So we know about some of these interleukins. So that's a cool thing to sort out in the future. And then the question is, what this altered metabolites situation, which we talked about, what is it doing to the viral infection in the lung? They didn't really look at that here, but are the mice better off when this happens, right? I think that would be really interesting to look at. The pathology, is it reduced somewhat? I'm sure they're working on that, and we're going to see that in the future. Because as Michele said, how cool would that be if part of our immune system, right?
Yeah. These are such important questions to human health, but my God, is it complicated? We've got a virus in the lung affecting the microbes in the gut and having an impact on certain cytokines and T cells.
Yeah.
And behavior.
Yeah, and behavior. That's right. But Helen Groves, who was the senior author on this paper, was actually, I think, a perfect match for this project. She said she always found it hard to choose one area of research to focus on. But she was lucky to be working with John Tregoning, because he too just has a really broad curiosity. But in her first year as a PhD student, she realized that she was really getting into something big here, because her thesis advisor entered her into a 3-minute thesis competition and gave the title, "What Does Flu Do to Your Poo?" So she said the next 4 years were lots of poo and mucus and mice, but lots of jokes and camaraderie, and working with the whole team she really enjoyed. Now, before she got into this project, how did she even get interested in science in the first place? Turns out both her parents and her brother were scientists. So she feels like in order to just keep up at the kitchen table, she learned pretty early on she better have data-driven arguments, because otherwise she wouldn't be able to be taken seriously. But she said she's always loved problem-solving detective work and is just fascinated by why some people get sick, why certain pathogens cause pandemics, others don't. She also appreciated with this particular project that she had 2 co-supervisors for her PhD. So they teamed with Mike Cox, who's a microbial ecologist, who kept trying to tell her it's not just groups of bacteria in the microbiome, it's actually a connected ecosystem. So that's something she that was imprinted on her through her advising from Mike Cox. And then also Professor Miriam Moffat was a respiratory immunology expert. So this also was just really important in trying to tackle this very complex but important biology. So when she reflects on her time as a PhD student, she said what she'd encourage other junior researchers to do is to try to find opportunities to augment your research training with other experiences where you're applying your science elsewhere. So, for example, she was able, during her graduate work, to also spend a year working in big pharma. She had her PhD in 2 very different labs, and then also did a 3-month internship at a charitable foundation supporting research funding. So she thinks this is a great way for trainees to really get a better feel for what opportunities are out there and also what really excites them. So now Helen is a full-time senior research officer for the Wellcome Trust, which is a nonprofit based in England. And she says her work now is to try to make sure that the research being done all around the world realizes its full potential to improve the health and lives of people. So she's worked on projects on cholera prevention and control, and also the impact of vaccines on fighting antimicrobial resistance, and then also epidemic preparedness. So what a great path she's had.
Nice. Very nice. Thank you, Michele.
Yeah.
All right, let me read just a few emails here to wrap it up. The first one is from BH, who writes, "Sorry if this is a misfire. I have a memory about a microbe that performs a sort of alchemy, shifting subatomic particles. Iron, lead, and gold were mentioned in the discussion. I thought I had listened to this reference in the most recent TWiM, but having just re-listened, I'm lost and confused. Does any of this sound familiar? I'm starting to think I fell asleep listening to TWiM and dreamed it." Michael, does that make any sense to you? Iron, lead, and gold?
It sounds like a recent TWiM, but I haven't candidly gone on and tried to figure out what episode it was from.
Okay.
Bacteria can effectively fractionate isotopes. We know that bacteria can actually fractionate different weights of sulfur. Some bacteria preferentially take up sulfur 32, sulfur 33, sulfur 34, sulfur 35. And so microbes are pretty discriminating in the weight of the neutrons associated with the isotopes. But I think we were talking about the alchemist's dream of turning lead to gold in that one episode where we were adding something. I'll have to go back and look. I think.
Do you remember the one where Elio did the story about the bacteria, the billion-year-old bacteria inside of the rocks? That
I think that was it. I think that was it.
Yeah.
Where I said it was the alchemist's dream.
Yeah. All right.
Of turning lead to gold.
All right, you've got your follow-up assignment, Michael.
Thank you.
Track it down.
P writes, to all the wonderful hosts, thank you so much. I really admire your deep knowledge, insights, and enthusiasm about various aspects of microbiology. I graduated from my PhD program many years ago and decided to choose a clinical path, but my heart never left the research field. Your shows not only helped me to reignite my passion about microbiology, but more importantly, brought me up to speed on what is current in the field. Science advances so quickly. There's so much to learn out there, so little time. It's so useful to have experienced mentors like yourselves to point us in the right direction, particularly selecting papers with good qualities to read and understand. I am a pediatric dentist who deals with caries all day, every day. It's disheartening to see little children suffering from this oral dysbiosis, and I'm only being reactive about it, i.e. doing restorations. I hope there will be advances in oral microbiology soon that will allow us to be more proactive for the disease. Your shows provide inspiration and hope that we will get there soon. Thank you again, and please keep up the good work.
We should do a dental paper, oral microbiota paper.
Yeah, that would be fun.
There's a lot of really neat stuff that's been coming out of late.
I'll bet.
Maybe you could find something for next time.
I'll go hunting.
We had Michael on TWiM, Michele, some time ago to talk about dentistry in the time of COVID.
Yes.
Right. Yeah.
That was fun. I got a lot of nice emails from folks out there, dentists who have done things. And, dentistry has had a rough time dealing with COVID because they generate aerosols and, they have figured out solutions to some of their problems.
I went not too long ago for a checkup, and it was fine. The only people there were the hygienists. The dentist was nowhere to be found. He was not having anything to do with it. But the two ladies were there, and they were cheery, and everything was fine. It was great. And
I hope they were vaccinated too.
I'm sure they were. Yeah.
Well, right now, our pediatric dentist's only solution is to literally coat the teeth with plastic, sealants to effectively keep the microbes from making the acid to disrupt the hydroxyapatite crystal. And of course, the safest thing you can do is make sure you have fluoride in your water, because that changes the equilibrium constant of the hydroxyapatite disequilibrium secondary to acid.
Yeah. All right, that'll do it for TWiM 241. You can find the show notes at microbe.tv/TWiM. If you have a question or a comment, we'd love to hear it. TWiM, TWIM at microbe.tv. And if you really like what we do, you could support us financially. Go over to microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you. Nice to see you both.
Michael Schmidt's at the Medical University of South Carolina. Thanks, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.